So in this lecture we'll be seeing about diabetes and its pharmacotherapy. So when we want to know about the pharmacotherapy of diabetes, it is important that we understand about the pancreas. So first we'll be seeing about the pancreas, and then we'll be seeing how the insulin is secreted and what stimulates the insulin secretion, its packaging and the mechanism of action of insulin, and the drugs which are used in the treatment of diabetes mellitus. So that is how this lecture will go. So first we'll be seeing about the pancreas. Okay, so the pancreas has two functions. One is the exocrine function, which constitutes almost 99% of the function of pancreas, and next is the endocrine portion, which constitutes 1% of the function of pancreas. So we should know the cells which are doing the exocrine functions and the cell which are doing the endocrine functions. So the exocrine function is performed by mainly two cells, that is the acinar cells and then the duct cells. So these are the main two cells which secrete trypsinogen, amylase and do the exocrine functions. But in this lecture we will be covering the endocrine function of the pancreas, which is important for the secretion of insulin and keeping the glucose in check. So the endocrine part has many cells, and you should know in detail about the cells which are involved in the endocrine functions. So these are the cells: alpha cells, beta cells, delta cells and F cells. So these cells are the main cells which are involved in the endocrine function, and each of the cells secrete a different enzyme. So each has a different function. So the alpha cells secrete glucagon, the beta cells secrete the most important insulin, and then your delta cells, they secrete somatostatin, and F cells secrete pancreatic polypeptide. So these four are secreted by different cells which are present in the endocrine portion of the pancreas. So clubbed together, these are present as islet of Langerhans in the pancreas. So this is the most famous term involved with diabetes. So now we'll see what the beta cells secrete. I said earlier that the beta cells secrete insulin, but it does not secrete only insulin. So what other things does it secrete? It secretes insulin, it secretes the C peptide, it secretes some pro-insulin also, and most importantly it also secretes amylin. So that is one important thing you should remember while studying diabetes. So these four are secreted by your beta cells. So now we'll see how the insulin is secreted. That is the first step we'll be seeing in this lecture. So now coming to the insulin packaging. The first thing we should remember before going into the packaging is that which chromosome is responsible for the secretion of insulin. So which chromosome? The chromosome which is involved is the chromosome 11. The short arm of the chromosome 11 is responsible for the secretion of insulin. It contains the information which codes for this. So from this the mRNA is synthesized. So this mRNA will contain the genes or the information which code the insulin. So this comes out to the cytoplasm, where the ribosomes act on the mRNA and they lead to secretion of what is called as the signal protein. This is the first step in the insulin packaging. That is, from the chromosome 11, that is the short arm, you get a mRNA with all the information which is acted upon by the ribosomes, and a signal protein is formed. So next, where does this signal protein go? It goes to the rough endoplasmic reticulum. So this signal protein is inserted into the rough endoplasmic reticulum. So what happens when the signal protein is inserted into the rough endoplasmic reticulum? So it undergoes elongation. So this signal protein will undergo elongation. So what will it be elongated into? What will the signal protein be elongated into? So this is called the pre-pro-insulin. This is the first major step in the synthesis of insulin. A pre-pro-insulin is secreted from the chromosome 11 into the rough endoplasmic reticulum. So we need to know the structure of the pre-pro-insulin. So this is the structure of the pre-pro-insulin. It contains 110 amino acids. So remember this number. So it contains of 110 amino acids. So whenever you say pre-pro-insulin, 110 amino acids should come into your mind. So 110 amino acids. So this is the signal protein. You have your B part and then C peptide and a part which codes for the A subunit. So this consists of 24 amino acids, whereas the B part, it consists of 30 amino acids. This C part consists of 31 amino acids and the A subunit consists of 21 amino acids. When you count the amino acids, the count will come to 106. So I said that the total number of amino acids is 110, but when you count this, the total comes to 106. So where are the rest 4 amino acids? They are present here. This is PC1 and PC2. This contains of 2 amino acids and this contains of 2 amino acids. So the full form of this is prohormone convertase enzymes. So this codes for the prohormone convertase enzymes, which I will say later step where it is involved. So these lead to the 4 amino acids. So now the total comes to 110 amino acids. So now you know the structure of the pre-pro-insulin, right? So it has 110 amino acids and it has coding regions for the signal peptide, the B subunit, the C subunit and the A subunit. So now the next step. The next step, there are endonucleases which are present in the rough endoplasmic reticulum. Endonucleases. So this endonucleases will act on the pre-pro-insulin and they will cleave the signal peptide from the pre-pro-insulin. That is the next step. So endopeptides will act and they will cleave the signal protein. So you have your B subunit, C subunit and the pro-convertase enzymes and the A subunit. Then there are signal peptides which are formed. So next, from this pre-pro-insulin, your signal protein is cleaved off and then your pro-insulin is formed. So the insulin is packaged properly and there is formation of disulfide bridges and which is called your pro-insulin. So now you have a product called pro-insulin which has a subunit, your C subunit and which are attached by disulfide bridges. Disulfide bridges. Okay. So now this entire pro-insulin, it escapes with the disulfide bridges, it escapes into your Golgi apparatus. So this is the Golgi apparatus. So this escapes into the Golgi apparatus, this pro-insulin. So now the action of the prohormone convertase enzyme comes into action in the Golgi apparatus, where this PC1 and PC2 acts and then the pro-insulin is cleaved into A and B peptide with the disulfide bridges and the C peptide. So this entire part with the A subunit and the B subunit is called your insulin, and the C peptide is present separately, and there are some insulins which remain as it is, so which are present here. So this entire thing is secreted here. So now you have a secretory granule which consists of an insulin which has your A subunit and B subunit with disulfide bridges, which has your C peptide, and which has some unconverted proinsulin. So this is the secretory granule. So now you have packaged your insulin. That is the first step in the secretion, right? So what else you need to know here is that when the insulin is secreted, it is kept in conjugation by two zinc molecules, and the insulin is secreted as a hexamer. Okay, so this is what you should know. The two zinc molecules help to keep it in conjugation in a hexamer state, right? So now we have seen how the secretory molecules are formed. Now we need to know what causes the secretion of the insulin from the secretory granules into the cells. So that is what we are going to see now. So we all have heard about the glut receptors, right? So this causes glucose to enter into the cell. So what happens when glucose enters the cell? So it is acted upon by glucokinase, which converts it into glucose 6-phosphate, which is converted to pyruvate, which enters the TCA cycle. This happens in the mitochondria, which enters into the TCA cycle, and oxidative phosphorylation takes place. And this leads to conversion of your ADP into ATP. So normally you have ATP dependent potassium channels here, which are always open, okay, and then you have calcium channels here, so which are generally closed. So when this ATP goes and binds to this channel, what it does is that it closes the potassium channels and causes opening of, which causes depolarization and causes opening of the calcium channels. So when the calcium channels are opened, so secretory granules consisting of your insulin, your C peptide, proinsulin are present here. So this causes fusion of this secretory granule here, and then it causes release of insulin, right? So we will revise the entire step. So when glucose comes into the cell, it is acted upon by glucokinase and converted into G6P, which is converted to pyruvate, which undergoes the TCA cycle in the mitochondria, which results in formation of ATP. So this ATP goes and blocks your K plus channels, leading to depolarization and entry of the calcium ions, which acts on the secretory granules and leads to secretion of your insulin. So now we will be seeing about the potassium channels in detail. So why do we need to know about the potassium channels is that there are a group of drugs called sulfonylureas which act by closure of the potassium channels. So we need to know how it does that. So now I will be zooming in into one of the potassium channels here. So this is how a potassium channel looks like. So the inner structure, so it is a tetrameric structure. So the inner structure consists of KIR 6.2, 4 subunits, so which are linked to the sulfonylurea receptors called SUR1. Okay, So these two things you should remember. So what is KIR, you might be wondering. So that is the potassium inwardly rectifying 6.2. So now this is coupled to the SIR1. So now what does the SIR consist of? It consists of 3 transmembrane domains, that is TM0, TM1 and TMD2. So what does TMD mean? TMD means transmembrane domains. So this consists of 3 transmembrane domains, that is 0, 1 and 2, and it consists of 2 loops, the nucleotide folding loops. So it has 2 loops here. Okay, so this is the nucleotide binding loop 1, nucleotide binding loop 1, and this is the nucleotide binding loop 2. So what happens here? So this ATP, which we saw in the late stage here, this ATP gets attached to the nucleotide binding loop 1. So it causes a conformational change in the SUR. So this causes the closure. So here there is a pore. So it causes conformational change in the KIR 6.2. So what happens? This pore which was open and it was causing the entry of your potassium ions will now close, and there will be no entry of your potassium ions. So this is how this channel acts. So the in-depth knowledge is necessary to understand your sulfonylurea drugs also. So this is how it works. So now in the next step, now we have got insulin, right? So now we'll be seeing how this insulin goes and acts on its receptor and how it shows its action on a cell. For example, you imagine this is a cell, okay, where you have a insulin receptor. This receptor, the insulin receptor, consists of 2 alpha subunits and 2 beta subunits. So imagine these are the alpha subunits and these are the beta subunits. So the alpha subunits and the beta subunits are connected again by disulfide bridges. So the first step, when the insulin is secreted there, this insulin comes and binds to the insulin receptor. So this insulin receptor is a kind of tyrosine kinase receptor. So we know that there are 4 major kind of receptors: the inotropic receptors, G protein coupled receptor, tyrosine kinase receptors, and metabotropic receptors, or the nuclear receptors. So now this receptor is a tyrosine kinase receptor. So now what happens in the first stage itself? There will be auto phosphorylation of these receptors. So that is the first step. First step is auto phosphorylation. Okay. So when the insulin comes and binds to this receptor, there occurs a autophosphorylation here. So what happens when autophosphorylation takes place? There should be some activation, right? So here what get activated? Insulin receptor substrate 1. So this is the major substrate which gets activated by the activation of your insulin receptor. So this insulin receptor substrate 1 also gets phosphorylated. So what happens when the insulin receptor substrate 1 is phosphorylated? That we should know in great detail. So first it leads to activation of GRB2 here, and then it leads to activation of PIP3. Okay. So GRB2, if you see my lectures on the tyrosine kinase receptor, you will be knowing that GRB2 is the first step in your MAP kinase pathway, and PIP3, this is a pathway called PIP3 AKT pathway. And then there is also formation of your IP3 and DAG, which leads to activation of your PK. So what happens due to activation of all these pathways? So the activation of PIP3 pathway leads to cell survival. This MAP kinase pathway leads to cell proliferation. Now the activation of this protein kinase, which is a protein kinase C, will lead to increased glucose uptake. It will lead to increased storage of glycogen, and it will decrease the breakdown of glycogen. And then this PIP3 AKT pathway will also lead to formation of the GLUT4 receptors, which are present in the vesicle, and they are placed on the surface of the cell. So now we will recap the entire function, what the insulin does in the cell. So now the insulin will bind to its receptor, it will get autophosphorylated. This phosphorylation will lead to phosphorylation and activation of IRS, that is your insulin receptor substrate 1. So that will get phosphorylated, and it will lead to first activation of the GRB2, which will further activate your MAP kinase pathway and lead to cell proliferation. And then it will lead to activation of PIP3, which will lead to activation of the PIP3K AKT pathway, which will lead to the cell survival. And then there will be secretion of your inositol triphosphate and diacylglycerol, which will lead to activation of the protein kinase C. This protein kinase C activation will lead to increased glucose uptake, increased storage of glycogen, and decreased breakdown of the glycogen. And also this PIP3 AKT pathway will lead to the formation of GLUT4 receptors, which will be embedded in your cell. So this is the entire function of the insulin on the cell. So next we will see about when you take food. The GIT releases certain substances which regulate the insulin secretion. These substances were identified as incretins. The incretins include glucagon like peptide 1, gastric inhibiting peptide, CCK, and other hormones. So we will be seeing about the GLP-1 and GIP in great detail. First let us see about GLP-1. The GLP-1 gets attached to its receptor, which is a GS type of receptor, right? So what is a GS type of receptor? It belongs to the G protein coupled receptors. So it will lead to activation of the adenyl cyclase, which leads to the formation of the cyclic AMP. This cyclic AMP will lead to increased calcium. So what is the function of this GLP? What does it exactly do is that this increased calcium, which we saw earlier, the opening of the calcium or intrusion of the calcium into the cell will cause the secretion of insulin from the granules, right? So this is how the GLP-1 causes secretion of insulin. That is the first thing. So it increases the insulin secretion. Okay. The second thing, it reduces gastric emptying gastric emptying time. So that is the second function of the GLP-1. And then third, it increases satiety. What is meant by satiety? So that is the lack of hunger. So you don't get hungry and it leads to decrease in obesity. First is it increases insulin, next it reduces gastric emptying, and it increases satiety. So a good doctor should know these steps in detail, and a very good doctor should know that it also leads to decrease in apoptosis. So how does it reduce apoptosis? It leads to this increased CMP will lead to increased CREB, that is your cyclic AMP responsive element binding protein. So this increased cyclic AMP will lead to increase in CREB. So this will lead to increase in BCL2. So this will lead to decrease in the function of caspase 3, and ultimately there will be reduction in apoptosis, and there will be also be decrease in the oxidative stress. So these are the 5 main functions. So first is it will increase the insulin, it reduces the gastric emptying time, it increases satiety, it decreases apoptosis, and it also decreases the oxidative stress. So these are the functions of the GLP-1. And then the GIP also has similar functions, but the half-life of the GLP-1 is very less, and it gets immediately degraded by an enzyme called DPP4, dipeptidyl peptidase 4. So this leads to the metabolism or destruction of the GLP-1. Okay, you should remember this DPP4.